Thank you very much for the amazing opportunity to be here today. Thank you to the FDRS for the kind invitation, and of course to the Lipedema Foundation for the support. And yeah, so my name is Nono Nankan Pamela, and today I'm going to briefly talk about my research based in Leipzig, Germany. So adipose tissue is one of the major or larger organ in our body with an exceptional capacity of expansion. And it is like divided in two major depots, which is the white adipose tissue. That's what we generally mostly know, which is responsible of storage and cushioning the organs as well as endocrine signaling. But we also have brown adipose tissue, which is mostly present in early stage in life, like in babies, but also to some extent in adults. It's mostly responsible of thermogenesis, energy expenditure as well. So my research mostly focused on white adipose tissue in the context of lipedema as well. So in the time of high energy availability, adipose tissue expand in two major ways that have been described. So the first way is by adipocyte hyperplasia, which just means the increase in cell number. And it's supposed to be the good way how adipose tissue can expand. But on the other side, adipocytes or the fat cells, the major cell type in adipose tissue, which are adipocytes, can also expand mainly in the size. And this has been described mostly in obesity, as responsible of a lot of dysfunctions described in adipose tissue diseases. You may have guessed, that's right. So my research focused on adipose tissue in the context of obesity and also now lipedema. What I study mostly is the tissue structure, composition, going from gene expression, protein expression, all the way to single cells. And also, I study some tissue function. But of course, I also look first at the serum samples, also because it's just easy to access. And 2 years ago, it's already 3 years actually, we could show that compared to individuals with obesity, individuals with lipedema were characterized with higher level of HbA1c, which is the clinical evaluation of kind of insulin sensitivity, a bit easy measurement of the insulin sensitivity level. So this means that the individual with lipedema tend to be more insulin sensitive, as we heard in the talk just a few minutes ago. But we also could see down there, you see in red, lipid peroxidation. It's just showing that there is a higher level of oxidative stress. And in the blood, we also saw some high level of inflammatory markers. So that points us towards adipose tissue. And I hope you can appreciate, if it's clear enough, that at the bottom we have lipedema adipose tissue and the controls that were matched for BMI. So these are the controls with obesity. And we could see that the tissue or the cells coming from the lipedema tissue are bigger than the ones from obesity, and that has been shown before. I'm not going to ask you to focus on any small, tiny points there, but the point here is that in purple, you see those are inflammatory markers, and we saw that they tend to be down-regulated in the tissue of lipedema, of patients with lipedema. And we saw in red or in pink is rather an upregulation of thermogenic marker. And this was quite surprising to us, but now this is kind of backup. This is not published yet, but it's now back up with a recent publication that showed just about the same thing. So as I mentioned at the beginning of this talk, there is two major types of adipose tissue. The first one is white adipocytes that is generally responsible for fat storage, but then we have the beige adipocytes also, and the process of browning is known as like an intermediate phase between white and brown adipose tissue. So our result tends to suggest that there is a process of browning in the lipedema tissue. And this is, like UCP1 is the major molecule that is measured, like how we can measure that browning. And I'm just showing here the black bar, the UCP1 expression in lipedema, and you can appreciate how higher it is in the lipedema tissue. So the implication of this browning in lipedema definitely have to be further investigated, but it can be the reason why the women with lipedema tends to have a high insulin sensitivity and low glucose level. So I also just want to head up again about this estrogen research. It's definitely something that has to be more like implicated and involved even in the data collection. This is just, I realize that the studies that are available don't really study this, let's say in the study design, we tend to forget to match for, like take the information about their cycle and like this hormonal related stage. And I think this is very important that we start including it in the studies, because then it will help us to be able to further study the estrogen metabolism, which might be associated to lipedema onset and development. So in my studies, I also now look at the single cell sequencing. So let's say the adipose tissue is composed not only of fat cells, but also immune cells and a lot of other cells. It's a little bit like that bowl of fruits with a lot of like different, very delicious fruit in it. But what the bulk RNA sequencing, the general sequencing that we do, give us that smoothie. So we have like the average expression, but we cannot tell which cell is, the gene expression is coming from which cells particularly. Therefore, we cannot really know which cells we should target. And this is what the single cell help us to do. So we don't just get the general gene expression, but we also get which cells is secreting those genes and how the cells proportion differ. And therefore, that's what I did for lipedema, on lipedema tissues. And every group that you see different colors there are cell types. So I sequenced about 100,000 cells. And what I saw so far is that the adipocytes, the fat cells, appear to be more like higher proportion in lipedema, which make kind of sense. But even more interestingly, I realized like vascular cells tends to be lower in proportion in the lipedema tissue. Immune cells tends to be lower also, and the precursor cells, as Ashok just presented, tends to be more in lipedema. So here is just showing in a bigger cohort that vascular cells appear to reduce in proportion, maybe because the tissue is expanding so much, and the vascular cells are just not developing enough to be able to top up the adipose tissue development. Immune cells like T cells in a bigger cohort are also reduced. And finally, we now study also the adipocyte function. The point here is to see if the cells are able to release the lipids as normal or adipose tissue with obesity. And what I realized here is that the adipocytes from lipedema tends to release less lipid. Maybe that's why there is this difficulty to lose weight once they gain it. And I could see it on the cellular level. So yeah, at the end of this, I would just like to highlight that lipedema tissue seems to be composed both with very large cells and very small cells. And the implication of thermogenesis needs to be further studied in this condition, and as well as the implication of estrogen and estrogen receptors signaling pathway. Adipocyte seems to increase in proportion and vascular cells seems to decrease in proportion. Of course, this study is not just me. This is the PI, co-PI on this study, the director of my institute. All my colleagues, I would like to thank again the Lipedema Foundation for supporting part of this research, and a big, huge thank you to all the participants, the people with lipedema, and also the obesity donors, and you for your attention. Thank you.